Yeah, thank you, Yasmin, for the nice introduction. As mentioned, I'll be talking about a deep metric learning tool that we developed at Single Round for cell type annotation. Before I talk about MapCell, I would like to give a brief overview of what has been happening in the field of single cell. Over the last decade, there's been a lot of developments and innovations in the field of single cell. As you can see here, a study early in the 2010 has been able to publish only 10 cells or few hundred cells, whereas now the same study could publish a few thousand cells. So there are new techniques and protocols being published very often now. And with the cost of sequencing becoming cheaper and cheaper, we are able to generate a lot of data, meaning a lot of single cells. And we are able to answer a lot of interesting new biological questions, and in general address different aspects of the biology. Given that we have the capability to generate a lot of data, what is the purpose of cell annotation? Here is an example of a 10x PBMC data where it has gone through a standard workflow where the low quality cells have been filtered out and clustering has been performed, as well as the data has been projected on a two-dimensional UMAP. But with just this information, you're not able to get much insights into the biology of what is going on in the data. Once we perform cell annotation using the well-established gene markers, we are able to see that there are these clusters of cells like T cells that group together, and there are B cells and there are monocytes in this data set. And from this information, you can also gain the knowledge that the T cells are more abundant in this data set compared to the least abundant cell types like platelets and dendritic cells. So just adding the information of cell annotation gives you a lot of information from which you can gain a lot of biological knowledge. So in order to perform cell annotation, there are two ways. Manually, you use well-established gene markers from literature, which is the gold standard process for cell annotation. And you look for the expression of these markers in your data set and annotate them manually. However, given the amount of data we deal with nowadays, it is a labor-intensive process and a time-consuming one. And also, it is very subjective. Like different person looking at the same markers and annotating the data might come up with different set of annotations. So in order to avoid all this, we could use a tool that does it automatically for us. So when you go down the path of automated annotation, there are two different types. One is a marker-based, meaning the tools use the well-established markers from literature and annotate your query data set. The other method is using a reference-based data set where you need a single cell data set with cell annotation labels, and making use of this reference data set, the tool learns the pattern from this reference data set and transfers the annotation to your query data set. Here is a table from a review paper that shows some of the tools that are publicly available and the approach that they have taken. For example, for marker gene-based tools, there is this SECatch and Sina. And for reference-based tools, there are SingleR and SEMatch. However, these tools have certain limitations. For example, when it comes to marker-based tool, you are limited by the number of markers that is available. And most of these markers come from bulk-seq analysis and not really from the single cell data. And some of the tools require a lot of data for them to be trained and then to perform annotation. And given that they require a lot of data, this is not really applicable for rare cell types. If you have a rare cell population in your data, and you want to train a model, most of these methods might not be applicable. Given the limitation and accounting for these limitations, we developed a tool called MapCell. And the tool is based on an approach called similarity learning or metric learning. As the name implies, it is based on measuring the similarity of the cells, or in general, a similarity between two objects or two data points, instead of classifying the output. And the way it's done is by training an algorithm to learn a distance metric to perform the task. So the distance metric could be, the distance could be any distance that fits your data. It could be a Euclidean distance, or it could be a Manhattan distance or cosine. And the task here is whether to classify two different cell types, or disease versus normal, or in general, it could be cats versus dogs, or animal versus non-animal. So it is important to set the task, and depending on the task, you train the algorithm to learn the distance metric. And also MapCell follows a few short learning approach, meaning that the tool doesn't require a lot of information, meaning a lot of cells to be trained. And it is a supervised method, meaning it requires reference single cell data with proper annotation of cell types. And you can see the application of this metric learning based approaches in various fields, such as, since it's a distance based approach, it is applied for finding nearest neighbors, information retrieval, ranking, face detection, face verification, and character recognition, and so on and so forth. So it has a lot of applications in various fields. So our approach for the MapCell is, number one, we build a neural network to obtain a reduced embedding space or vector space. Given that we have 30,000 plus genes from the single cell data, we want to reduce the feature space to a reduced embedding space. And for this, we use a neural network to do this. And here is an example of a neural network architecture, which has an input layer of 30,000 plus genes, and two hidden layers with size of 512 neurons, and producing an output of 32-dimensional vector space. After we build the neural network model, we train the algorithm. We train the algorithm to using a loss function that is based on a metric learning. So there are several loss functions that are available based on metric learning, such as contrastive loss, quadruple loss, and one of them is triplet loss. So what are triplets? So triplets, as the name implies, it's composed of 3 units, and those 3 units are anchor, positive, and negative. So the anchor and positive belong to the same cell class, and the negative should belong to a different cell class. The purpose of this is, I'll explain in the next slide. So if you have a sample data set, the way it's created is the anchor and positive should belong to the same class, and the negative from a different class. So the reason for constructing triplet in such a way is that, since it's metric learning based, the program tries to bring, by learning from the embedding space, the anchor and positive together, since they both belong to the same class, and pushing away the negative cell. Here is a mathematical notation for the triplet loss function, where it takes in the distance between the anchor and positive, and then the anchor and negative, and then it calculates the loss function. So we want, in general, the distance between the anchor and positive to be lesser than the distance between an anchor and negative. And we add a margin to the anchor and positive distance, such that the negative slide further away from the positive cells. So depending on where the negative is in relative to the anchor and positive, there are 3 types of triplets. One is the easy triplet, second one is the semi-hard triplet, the third one is the hard negative triplet. So easy triplet is it's very distinct from the anchor and positive, whereas the semi-hard, it's similar to positive, but the distance is smaller than the anchor and positive. Whereas the hard negative, it's highly similar to a positive. So when you train your model, it is important to choose the correct triplet and train your model. Here is an example in the embedding space where initially, before you apply a triplet loss function, it might look like this, where the anchor and positive are too far away, and the anchor and negative are much closer. Once you apply the triplet loss function and train your neural network, the embedding space should look like this. So at each iteration, the algorithm computes a loss value, and based on that loss value, the weights of your the nodes in your neural network are adjusted. So here is a workflow of how it happens. Given that you have a data set consisting of different cells, the color represents different cells. We split them into batches and put it through a neural network architecture that produces an embedding space. And that embedding space is used to create all possible triplets. And based on this, a loss value is calculated. And based on this loss value, through back propagation, we update the neural network weights such that it achieves a shorter distance between an anchor and positive and a larger distance between an anchor and negative. So to train our MapCell model, we do perform hyperparameter tuning. So tuning different parameter in the model, the purpose of tuning different parameter in the model is to control the learning process. The purpose here is to find the right combination of parameters, such as to achieve a maximum performance or the best performance. In order to fine-tune some of the major parameters in our model, such as the number of nodes in layer 1, hidden layer 1 and hidden layer 2, as well as the final layer, or even the batch size, we use a tool called weights and biases, which performs hyperparameter tuning, and where we are able to track how different combination of parameter leads to reducing the loss on the validation data set. So based on this, we obtain an optimized model. Subsequent to this, we use the program called KNN, K nearest neighbor algorithm. Taking the embedding space from the trained model, we feed that to the KNN algorithm to predict our cell type. And there are different algorithms that are available for this purpose to find the nearest neighbors. One that we use is the scikit-learn KNN algorithm. But if you have a big query data set, if you have a big reference data set, or if your query is too big, you can always use alternative algorithms that are developed by Facebook called FIES, and even Google has some vector-based similarity search algorithms. So here is the overall workflow of MapCell, where you train, where you feed the input data, which is your query data, and feed that to the trained neural network, and you obtain an embedding space. And using that embedding space, you do perform cell type prediction. The embedding space can also be used for visualization purpose. So here is an example of a MapCell model that we developed using bone marrow data. So the data was obtained from this publication where they have collected cells from 20 LT individuals. And this is a 10x data, and the data is publicly available. So in this data set, they have annotated the cells, and then they found 19 different cell types to be found in the bone marrow. So for training of our MapCell, we use 30 cells from each of the cell type, and for validation, we use 10 cells, and the remaining we use to perform to measure the performance of a model. Here is a visualization plot where on the left you see an embedding space that was obtained before training the model itself by just using the neural network. If you were to feed all 30,000 genes and get an embedding space, this is what it might look like if you project it on a three-dimensional UMAP. But once you train the model with a triplet loss function, you are able to see that it is able to push cells that are different from further away and similar cells are brought closer together. So this is on the training data. So you can clearly see that it is able to separate different cell types. So next step, we apply this on the test data. Here is a subset of test data that we have applied on. And you can see that the cells are colored based on the ground truth here on the left, and on the right, it is colored based on the predicted cell type. Based on what the program has learned, based on the cell annotation provided, the program is able to predict to a greater accuracy here. And two things could be happening here. One is that some of the cell types look closer together because that the input fed to the model, which is just the RNA itself, is not sufficient to discriminate these cell types. And it is known from literature that some of the T cell subtypes are hard to distinguish just using the RNA. And the model also relies on the ground truth. So the ground truth has to be very good for the model to perform well. So if you look at, in order to quantitate the prediction accuracy, here is a confusion matrix. On the x-axis is the predicted label, and on the y-axis is the true label, which is the ground truth. And you could see that the program is challenged or it's struggling to classify some of the T cell subtypes. And as I mentioned, it is known from literature, it is you need protein information to discriminate some of these features, some of the cell types. Here is the performance metric values for each of the cell type. And you can see that we have an overall F1 score of 0.82. And for majority of the cell types, we have a good F1 score, except for the T cell. Again, we compare our method against some of the publicly available methods, such as single R and SEMAP and so on, where we use the same number of training cells that was used for our tool and compare against the other tools. And you can see that MapCell is able to outperform the tools shown here. And we show a metric called Matthews correlation coefficient, which is a metric that is used if you have an imbalanced data set, meaning the number of cells are not the same from different cell type. And you can see the score is 0.8 in comparison to all the other tools. MapCell is able to do well. So we have also tried developing a model using RNA and ADT, and it has shown that the accuracy of discriminating our T cell has increased, and we have an overall score of 0.9. Here is a use case where we looked at a disease-associated single cell data, and we found that there is a group of cells called C26 colored in red here. And these cells were found to be associated with prognosis of the disease. So we wanted to test if MapCell could be trained to predict this particular cell group with a good accuracy. So, and you can see on the left here, before training our model, it is not able to discriminate. Once we train the model based on the similarity learning loss function, it is able to discriminate the cells from the rest of the cells quite nicely. And we get an F1 score of 0.94. So we have shown that we show that MapCell is able to do quite well if you have very low number of cells and you want to do a prediction on this low number of cells. So in summary, the limitations of MapCell is that you need a model for each disease that you want to look at or each tissue that you want to look at. So obviously, since it's a reference-based model and it's a model that is trained based on the neural network, you need a model for each disease that you are looking into. And in terms of interpretability, it is very hard to interpret in general, just the deep learning neural network. And given that we are getting an embedding space of 32 dimension, it is much harder to interpret. So if you're looking for a model where you want to know the contribution of a particular gene to the outcome, it is impossible with MapCell model. And the advantages are many. As I said again and again, it requires very low number of cells, and with the low number of cells, we have proven that it could achieve good performance. And the time taken to train this model is not a lot as well. And apart from the cell type prediction, it also produces an embedding space that could be used for different purposes in the space of single cell analysis. One is this embedding space itself can be stored in a database and used for further querying in the future. And this could be also used for visualization purpose, as I've shown earlier. And if a tool is looking for generating a cell-cell similarity matrix, the embedding space could be used for such purpose as well. And in terms of prediction, it is quite fast, and it can predict around 40,000 cells in less than 2 minutes. So that's all I have for MapCell. If you have any further queries, I'll be happy to take it and then address it.
Yes, thank you so much for the very interesting and comprehensive talk. Yeah, so we can start Q&A section. We already have some questions. Then I start with the first one. Is MapCell applicable to data generated from other platforms like 10x or others? Is this technology dependent or independent?
Yes, we have conducted some tests on it, and MapCell is technology independent. It can take in data from any platform like 10x or Singleron or any other platform that is available there, because it works on the raw data. So it's quite robust in terms of taking in the data and doing a prediction.
Yes. The next question is, what are the different MapCell models available currently?
So far we have models for bone marrow, and then we have a model based on PBMC data. So both of these models do come with just RNA only as feature, input feature, plus RNA plus ADT, which is a protein. So both for bone marrow and PBMC, we have this, and we are still currently developing models for other tissues and disease-related models. So if you have any specific model that you want to be developed, we'll be happy to do that for you as well.
Mm-hmm. It is also asked that, it is asked, in your reference-based method, how do you count if some cell types are not present in the reference data?
Well, as I said, it is based on the reference data. So whatever you feed, that's what the model is being trained on. But you can definitely use your trained model and again, just take the new cell type that you want to be added to the model, and it can be updated subsequently. So it's not hard to add new cell type to the model. However, it is able to predict only what it has been trained for, meaning the cell types that it has been trained for. And you could use a confidence score that we have, along with the prediction, we do provide a confidence score based on the normalized exponential distance of the neighbors. So using this score, you have to do a bit of a manual creation to decide whether is it really the cell subtype or cell type or not. So you could use the confidence score to decide if a particular cell type is the right cell type in your data set.
Mhm. So next question, although single cell analysis is a powerful tool, various issues have been reported, including batch effects and transcriptional noise due to the gene expression variation and mRNA degradation. How can these challenges be overcome?
In terms of MapCell, we have tested by using batch corrected data as well as unbatch corrected data as well. And then it seems to be very robust in terms of its prediction. So as far as MapCell is concerned, it's not affected by batch effect here. And obviously, batch effect is a common issue in the field of single cell. And there are a lot of tools that are available out there to address these challenges. I'm not sure if you're looking for any specific tool or anything, but there are so many tools that are available out there. And we also have, at Singleron, have developed a tool called MapBatch to address batch effect for this purpose.
Mm-hmm. Can MapCell be used to classify patients instead of cell types, such as healthy versus diseased, or drug responders versus not responders?
Yes, definitely. In our paper that we published earlier, which has used contrastive laws, and what I just showed you is an upgraded version of the MapCell. So we have tested it on disease versus normal, and yes, we can train MapCell to predict a disease versus a normal patient as well. Or it could be anything. It can be drug responders or non-responders. So it can perform anything.
Interesting. So with the machine learning algorithm, how do you handle sensitivity and accuracy and linkage with spatial information parameters?
Sorry, I didn't quite get that question.
Okay, so if you add some explanations here, we will definitely address this question afterwards. Does it require normalization for every disease, or is it inbuilt for users across targets or tissues?
I'm not sure if I understand the first part of that question, but the second part is for different tissues and targets. Obviously, we'll have to, we are working on it. We are working on building different models for different tissues and targets. As far as the normalization, I do not get what that actually means.
Mm-hmm. So again, if you add more explanation, what exactly you would like to know, definitely we'll address this question during this webinar or afterwards. We contact you and get back to you and for sure answer your question. Can you combine different data sets from two independent experiments? Often we look at cancer tissue versus healthy tissue. What about batch effects?
Yeah, as I mentioned earlier, MapCell is very robust in taking in the raw data and performing the prediction. So it can combine your data and just provide the raw data to MapCell, and it is robust in doing its prediction. But in terms of like other analysis like clustering or other things, obviously you need to account for the batch effect. So in terms of cell type prediction with MapCell, you don't have to do batch correction.
Mm-hmm. How robust is MapCell in terms of estimation of expression levels, definition of cell identity, identification of gene signatures, and analysis of spatially resolved data?
Well, MapCell, it cannot estimate expression level because the MapCell has been trained to do cell type prediction. It is not an autoencoder that can recapitulate the expression data from a training or reference data. So it's not built for that purpose, actually. It's only for cell type identification. And for cell type identification, we do provide a confidence score to say how confident we are in predicting this particular cell type.
So with machine learning, how do you handle a situation whereby a gene is expressed but not detected by the sequencing technology, and those attributable to biologically true absence of expression?
Well, this question seems more like a question for wet lab people, I guess. Yeah, but obviously there are a lot of autoencoder based models that I've tried looking into it, and on top of my head, I can't remember the tool name, but there are tools out there to address this particular issues as well.
Yes. So thank you again for the very, very nice talk. Unfortunately, we are running out of time, but there are a lot of questions left. Definitely, we will get back to you with the answer. And don't hesitate to contact us using the information giving on the website here, on the slide here, sorry. And have a great day. And thank you for joining us again. Bye.
Thank you.